Welcome to our Bonito Lab training video on CTAB chloroform DNA extraction from fungal tissue. I'm Julian Liber. This protocol produces high molecular weight DNA appropriate for qPCR, PCR, and genome sequencing with Illumina technology. Preparation of DNA for long-read PacBio or nanopore sequencing requires a more stringent protocol. Prior to collecting fungal tissue, we are going to use 1.5 milliliter conical bottom microcentrifuge tubes with 2x CTAB buffer. CTAB is a detergent which helps preserve and extract DNA. It is also a carcinogen, so any materials such as gloves and tips which are contaminated must be disposed of as hazardous waste. We use green racks and work on the left side of the fume hood for DNA extraction in the Bonito Lab. Bring your tubes to the biosafety cabinet, placing the rack on a paper towel. Flame-sterilized forceps or other tools used to handle the fungal tissue. Label the caps of the tubes prior to adding the tissue. Remove the fungal tissue from a plate or flask, getting as much tissue as possible without overfilling the tube. You may need to practice to take an appropriate amount. Remove a sterilized pestle from a foil packet without touching the grinding end. Push the cap back before grinding, then use twisting and crushing motions to pulverize the tissue. Have enough paper towel underneath the tube to catch any spills, and use more paper towel and water to wipe up any spilled CTAB and dispose in the hazardous waste. Place used pestles in 50% bleach to clean them prior to re-autoclaving in foil packets. Add 450 microliters of 24:1 chloroform isoamyl alcohol to each tube. When pipetting, use filter tips and pipette up and down once before transferring to the tissue-containing tubes. This reagent breaks down cells and releases the DNA. Replace the lid tightly and store the bottle in the fume hood or flammables cabinet. Shake each tube vigorously until it appears milky. Remove the rotor lid and load the tubes. When loading the centrifuge, make sure all tubes are properly balanced by counting the holes between tubes and using counterweights if necessary. Centrifuge at maximum speed for 5 to 10 minutes. After centrifugation, you should see DNA containing liquid on top, the fungal tissue in the middle, and the chloroform at the bottom. Handle tubes carefully to avoid disrupting these layers. Using a P200, transfer the top layer to a new labeled tube. Count as you go and use the graduations to measure. For the first one, you can almost always get the full 200 microliters. After you have finished transferring as much of the supernatant as possible without including any chloroform, write the volume transferred on the side of the new tube. Chloroform will appear as tiny droplets which are denser than the rest of the liquid. You take up from one tube, then transfer to the empty tube with the same label. Leave the tube with the remaining chloroform and tissue to evaporate on the racks in the fume hood. If the DNA is going to be used for genome sequencing, add 6 microliters of RNase A, 25 milligrams per milliliter, to each tube. Invert several times and incubate at 37 degrees Celsius for 30 minutes in an incubator or water bath. If not, skip to the isopropyl alcohol step. Add an equal amount of chloroform isoamyl alcohol to the volume written on the tube. Shake and centrifuge for 5 to 10 minutes. After centrifugation, you should see 2 layers, with the top layer containing the DNA. Transfer the supernatant to a new tube and record the volume. Retrieve 100% isopropyl alcohol from the freezer used for flammable solvents. Add 0.6 times the volume on the tube. If careful to not touch the tubes, you can use the same tip for all of your samples. Invert the tube several times before placing in the -80 freezer for 8 minutes in an open-sided rack. You can put the isopropyl alcohol back while this is freezing. After removing from the freezer, centrifuge the tubes at 4 degrees Celsius for 15 minutes at maximum speed. Take care to arrange the tube hinges to face up. This step will leave a precipitated pellet of DNA. Remove tubes from the centrifuge. You may need to tap the rotor lid to release it. You should see a pellet of DNA at the bottom of the tube. Pour off the isopropyl alcohol into a waste tube, which will later go into a liquid hazardous waste container. Add 800 microliters of chilled 80% ethanol found in the same freezer as isopropyl alcohol and centrifuge for 90 minutes at maximum speed. Pour off ethanol and repeat. Remove ethanol using a P200 pipette, and you can eject the tip with the liquid. This liquid will evaporate. To remove all ethanol, we will vacuum dry the samples. Turn on the vacuum pump at the power strip. Load the speedvac centrifuge with your samples, making sure that they are balanced using an empty tube if odd-numbered. Close the lid and turn the concentrator on. Turn the valve on top and check the lid. Wait 2 to 3 minutes, then return. Turn the vacuum pump off and turn the valve to release the vacuum. Turn the concentrator off and remove your samples without losing the pellets. Add 55 microliters of nucleus-free water. I like to warm the tube of water prior to adding it, using water warmed in a beaker or a vial in the microwave. You should flick your tubes to resuspend the DNA after an hour at room temperature or overnight in the fridge. Freeze the samples at -20 Celsius for later use or -80 C for long-term storage. Do not vortex. Dispose of gloves in the hazardous waste, remove your lab coat, and wash your hands thoroughly. You should make sure all wastes are disposed of and reagents are returned to their storage locations. Thank you for watching, and good luck on your extractions!